Introduction. So I'm often introduced as a biologist because we think in biology terms and trying to solve the biological problem of the biology of aging. Actually, I was studying as a chemist. I had an undergraduate degree in chemistry. This is the chemistry department of Moscow State University. This is physics department. See, it's asymmetrical from chemistry. This is a statue of Lomonosov, who is the founder of the university, and physics were our arch rivals. We measured who is closer, whether he is closer to the chemistry or to the physics, and interestingly, it depended on who measures it. But I love physicists. For the 25 years of my lab, I always had theoretical physicists in my lab, and they made the most important discoveries, some of the most discoveries in my lab. And actually, two former lab members who give talks at this conference are a physicist, Andrei, sitting here, and then Csaba, who is now a PI in Hungary. He's a mathematician, not physicist, but it's close. So this is an amazing conference. I really enjoying it, and I wanted to give a high-level talk without formulas or data. Actually, this is one would be unusual for me, and it's an interesting opportunity. And I want to talk about aging in the most simple ways, the most simplest possible ways, just trying to understand the nature of the aging process. To me, when we think in a very simple way, we go from here to here. Damage causes functional decline, causes diseases, causes mortality. And so for various people in this audience and in the field, when we say we study aging, we mean different things. Some people think that aging is here, some here, here, and here. For example, many speakers mentioned that aging is the increased chance of dying with age. To me, it's not, because mortality may be caused by other factors. So for example, when we consider like 1-year-old and 5-year-old child, who is older? Because 5-year-old child is less likely to die than 1-year-old, less likely to have diseases, but you know, it looks older than the 1-year-old. Or for example, males 20 to 25 years old, constant mortality, or neck more rats, don't show increased mortality with age. So to me, mortality is one of the manifestations of the aging process, not aging. In the same way for disease and functional decline, you know, like VO2 max and other features, you know, they can be modulated in a way, and they're also very different in different people. Whereas damage, I think, is a more defining feature of the aging process. But of course, it depends on the semantics in a way. Damage has been implicated in many models and theories of aging, whether we look at adaptive or non-adaptive theories like antagonistic pleiotropy or disposable soma, I don't know, mutation accumulation, programmed aging. So they all kind of consider damage as the defining feature of the aging process. But what is that damage? How is it produced? How the organisms deal with it? That's completely unclear currently. So, because I was trained as a homologist, I often think in terms of enzymes. Let's consider an enzyme. It catalyzes a reaction, chemical reaction, of converting a substrate to a product. This is a function, so enzyme is needed to support that function. But there is a non-zero likelihood that this enzyme will produce something else. It could be a byproduct, or it could be, it could react with something else or modify in ways some other process. It's a non-zero likelihood because nothing is perfect in chemistry, and therefore in biology it's also imperfect. So now we have these two features of that enzyme. One is positive, that evolved, right? It's an adaptive feature, function. And one is unavoidable consequence of having that function. So for example, if you consider antagonistic pleiotropy, it has a positive function, but also negative function later in life. So if you consider an enzyme, that you already have that from the very start, without invoking any evolutionary process. It's the nature of life in a way, of any function. It has a positive and cumulative negative function. So this concept can be expanded to any other function in organisms. And so therefore we have this damage in the form of byproducts or mutations or epigenetic drifts or errors, imbalance, miscommunications between cells and organs. It could be all other known and unknown forms of damage at all levels of biological organization. So some people call it like cumulative damage. In our lab we call it deleteriome. I think it's a better term because the cumulative damage kind of only encompasses kind of chemical damage, like mutations, byproducts, whereas we are talking about any negative consequence of having functions. So in the more physical terms, we could say, it's increase in entropy or disorder or randomness. So I would stress that all functions have these antagonistic pleiotropy features, and every function contributes to total damage, and there are none of the damage forms is major. So how do organisms deal with that damage? The whole biology is designed to deal with that damage. We have all kinds of features, detoxification systems, exporters, we have lymphatic system or renal system. But I think the most important one is damage dilution by cell division. That's the nature of life. From the very first protocells that they had to think how to export entropy, all this damage that is produced unavoidably, it needs to be diluted. So when we consider the origin of life, the first thing we should consider actually is dilution of entropy. And it's done by cell division. For example, in this cell, we have 2 damages. Cell grows, now it's 4, and then divides, now it's 2. So the cell does not need to know about that damage. Does not need to evolve a system that deals with it. It can evolve systems, many systems that deal with some of the damage forms, like the most critical damage forms, but it does not need to deal with all of them. Because with all of them, there are always more damage forms than ways to protect against it. But cell dilution deals with this problem already. I think that's the most important feature of life. So I will show you just a couple of examples of how we study damage, because it's very difficult actually, because it's so small and so many of these damage forms, it's unclear how we exactly could quantify it and how we can develop approaches to study it. So in this study published already 10 years ago, we studied flies. And with mortality of flies, this is like a mortality curve, and this is under calorie restriction. And in red, there's a number of chemical species that we can detect as a function of age of the flies. And you can see this chemical diversity increases and kind of stops later. And then this is under calorie restriction conditions. We see this consistently the same. So we think it's a tip of the iceberg. We don't know whether any of these increases represent like actual metabolites or damage forms, but because there is a number of increases, we think it represents in part this damages. So another experiment we've published 7 years ago. Here we study whether damage is causal to aging. So what we did, we have these young organisms, flies, yeast, or mammals, and we also have all the same organisms, and we use them to make diets. So for example, we grind young yeast cells or old yeast cells and use as a diet to grow live yeast cells. Or like young flies and old flies, we grind them to make diet to grow flies. And so we look whether this diet, which is enriched in the damages, would be shortening or extending lifespan. And what we consistently found is that when organisms live on the diet, on the young diet, they live longer than on the short diet. So we think this damage, and we try to match the diet and the organism, like 1-to-1. So I think we conclude that these age-associated molecular changes are deleterious, causally linked to aging, and then affect lifespan through diet. Another example is actually from Andrei. He published this paper last year. So we studied here the contribution of co-regulated and stochastic changes, because it wasn't clear. And I would say that Andrew already mentioned a paper. Actually, there are 3 papers back-to-back in Nature Aging, and Sebastian is not in here. So thank you very much, Nature Aging, for supporting these important studies. Basically, all 3 studies, they have similar conclusions. But in our case, we try to quantify this, and we'll actually find co-regulated changes, but also stochastic changes. So it kind of goes back to this idea of reversible changes, dynamic versus stochastic, which is already discussed. Now I'd like to tell a little bit about the difference between aging and longevity. And I use this metaphor to represent this. To me, the regulation of lifespan and conversion from aging to non-aging state and vice versa is orthogonal questions. And it's well represented by this metaphor. Consider a river that goes from the mountain to the ocean. Why does the river flow? It flows because of the gravity. So to me, aging is the equivalent of gravity in this model, whereas the time that the river flows is equivalent to longevity. We can modify this time. We could reroute the river. We could build a dam. We have all other kind of features to modify this. It's equivalent to how in biology we have biological innovations. We have detoxification systems, so all kinds of other protection systems, and we have genes and we have functions. This is the essence of biology. Okay, but the gravity is the essence of aging. It has a physical underlying process. Yeah? It's rooted in physics. So what about rejuvenation? Rejuvenation, I think it is possible. So again, so in this example, a river flows, that's aging, and we could slow down by various interventions, aging in this direction, but what we want to take water from here to here. So, and even though aging is irreversible, the aging process, because it's very complicated, so it's impossible to go back in the same direction, but we could reverse biological age if it's represented by the deleterial, by cumulative damage. Because if we can deal, the biology can deal with this damage, we can export it, we can maybe replace organ, there are various ways. And this way we could rejuvenate biological age. Biological age could go down, even though aging itself is irreversible. So what are the examples? We studied and we published a few examples of rejuvenation. One example is this, we call it embryonic rejuvenation, because with every generation, we have a germline, and germline a little bit ages. Like sperm from older man, a little bit older than from a young man, for example, same for oocytes. This means there must be a rejuvenation process somehow, somewhere. Otherwise, the next generation would be older. And so we find that we observe this in early development. It's approximately corresponding in humans, maybe about the third week of development. It's a stage of gastrulation. So, yeah, even though, of course, when we use biological clocks, aging clocks, then they are out of range in a way. So there are challenges in interpreting this, but we consistently observe whether we apply transcriptomic clocks, like epigenetic clocks, when we apply single-cell clocks, and conceptually we must observe this. So it's kind of self-consistent. At the same time, we see other changes, like for telomere extension and so on. So this illustrates that this process from the zygote, which is the fertilized egg, to ground zero, we call it ground zero, the lowest biological age, there is a rejuvenation process. And we don't know mechanistically what it involves. I think that there's a massive cell division here, so it may be contributing to this process. But we don't know for sure mechanistically how it happens. But I think it's of major importance. And if we understand this process, maybe we could use it in the future in adult cells, somatic cells. So another example is parabiosis. This was a paper published in 2023 also in Nature Aging by Bohan Zeng. At that time he was a graduate student, now a postdoc in the lab. And he used the parabiosis to connect mice for 3 months. It's a really long time in mice. It's about 10% of their life. And after 3 months, we separated the mice, and the mice become rejuvenated by any measures that we have, and they also longer lived. So to me, this is also a very robust process of rejuvenation, and it's actually very difficult to recapitulate it with, for example, injection of young blood or replacing organs. We try all kinds of conditions, but actually parabiosis seems to be the most robust currently. It's very difficult to rejuvenate adult mice. And I also wanted to mention this example. It's not exactly rejuvenation, but kind of relates to the questions that already been discussed on the dynamical biological age. And this is the Jesse Paganik cell metabolism paper. And we found that under severe adversity, like for example, major surgery, COVID-19, pregnancy, parabiosis, and some other examples, we see a reversible change in biological age. It can be conceptualized in terms of the damage. Under like severe stress, organism is unable to deal with this damage, and damage accumulates. And later, when stress is relieved, organism can deal with that damage. It goes down, and the biological age is decreased. So it can change literally on a short time scales, I think. So, and finally, I wanted to mention this issue of heterogeneity, because we often think of biological age as organismal biological age. But of course, organism is a complex system. It has different organs and systems and cells. And so you could say that maybe there are clocks in different organs. And of course, again, besides organs, there are also cells. And we study this. Alex Trapp, a former lab member, he's now at Retro, he developed the first single-cell epigenetic clock. And what he finds is that there is a huge heterogeneity in cells in the organs. So cells, they interact with one another. They kind of exchange entropy in a way, but only partially. So there is still diversity of biological ages in different cells. And another example is from the paper by Ludger Gumini, a recent paper where he was able to quantify biological age of organs in humans based on plasma proteomic profiles. And when we identify proteins which detected in the plasma but made, for example, in the brain, we could develop a clock that predicts the biological age of the brain. And this way we were able to tell that various people, they have different aging trajectories in different organs. And this is a kind of model. So many organs, they age in a coordinated way, but sometimes there are organs which show increased biological age. But it's not simply increased, because this organ still interacts with other organs. Other organs kind of pull back that biological age, so the organ becomes altered. So, and we don't know yet how to best describe that state of that organ. But so it's both older and altered. And these are the organs that will develop diseases and eventually, the cause mortality, and they are different in different people. So this also immediately means that probably there will be no universal intervention, or at least that such interventions will be very rare. Because many interventions, they would depend on, we need to identify who can benefit from these interventions, basically. So, so what are the insights that we learned from this? So first about aging. Aging to me, what is aging? Aging is the increased deleteriome, which means that deleterious changes at all levels of biological organizations. These are mostly chemical, because the aging manifests in a chemical way, in chemical terms in organisms. But of course, the basis of it is physical, like entropy and disorder. The aging begins at gastrulation, around ground zero, and I also think that organismal life begins also at that state. So the period before that is kind of cellular life, preparation for organismal life. Aging is rooted in the second law of thermodynamics. It's a default state. So it's like a gravity. It cannot evolve. It does not evolve. It has no drivers. It's there from the very start. That's aging to me. And but longevity is different, and biological organization is different. And so in terms of longevity is adaptive. So all these theories that are proposed, antagonistic pleiotropy, it deals with the longevity, with the question how long organisms live, not why they age. Because antagonistic pleiotropy properties, they are already from the very start, in every organism, from the very first protocells. Yeah? And life works by counteracting aging, basically counteracting biology. We have all these innovations. That's life, biological functions. It works to basically slow the increase in entropy to sustain order. And sometimes it may stop it. So we have organisms that presumably do not age, like Planaria and Hydra. So biological innovations are such that it's possible to stop growth of entropy. And sometimes it's possible to decrease entropy, and that's rejuvenation. What is rejuvenation? Rejuvenation is the decreased deleteriome. I showed you some examples of that. And cell reprogramming also, I think, represents rejuvenation. And also, it's not exactly rejuvenation, but aging can I think, fluctuate. Biological age can fluctuate at the short time scales, perhaps even during the day, so or maybe seasonally and so on. So biological age may change. Finally, I mentioned about this organ and system-specific aging, and it's actually at all levels of organization, from cells to organs and systems and the entire organisms. And because of that, biological age in some cells or organs, or maybe even like individual genes, increases, and it represents a disease. So to me, the question, for example, whether aging a disease is a nonsensical question. But the opposite question is right, whether disease is aging. And to me, disease is aging. It's just basically a localized non-autonomous aging. And so in that sense, that medical disorders, like diseases, can be best understood as critical levels of physical disorder in a particular system, whether that disease happens. But also, when we think about aging, I think aging has this physical inevitability. It manifests chemically and mitigated biologically. And we need to bring actually all these three disciplines together to best understand it. This is my lab, the current lab, and also former lab members contributed, like Chaba, for example, here, who discovered embryonic rejuvenation, and Andrei and others, and many collaborators and funding sources. Thank you very much.
Thank you very much. Yes, the floor is open for questions.
Okay. Yeah, so if I can go ahead. So I was very captivated by one of the parts where you show in the early embryogenesis, right, prior to gastrulation, you think there is actually an increased rejuvenation, or rather that's the part that's rejuvenation. And then post-implantation, then gastrulation, ground zero heats up. So can you elaborate a little bit more on the rejuvenation, like what really supports that part that is, you know, showing evidence that there's actually rejuvenation, or is this something more speculative? Sorry.
Well, there's some evidence. Again, we don't understand mechanistically how this happens. I already discussed that we must have a rejuvenation process. Otherwise, the next generation will be older because the germline ages slightly. It's well protected. Its biological age very low, but it ages just a little bit. So that means it must be rejuvenated at some point. So where is that point? So you have like older sperm and older oocytes from older parents, right? So now we have zygote, which is slightly older zygote of the older parents compared to the younger parents, but yet all organisms kind of begin life very similarly. So there must be a rejuvenation process. So now the question is, you know, how would you test it? And so we test with the available tools. So even though there are limitations of clocks, but we apply different clocks. So for example, we initially apply the Horvath clock, we observe rejuvenation. We apply the Hannum rejuvenation, Grimm-H rejuvenation. And we say, okay, how about we apply single cell clock? We develop a single cell clock, we apply, we observe rejuvenation. We develop transcriptomic clock, we observe rejuvenation. So we look at the damage, for example, telomere extension, telomeres are extended at the same period. So there must be rejuvenation, and we observe rejuvenation, even though any individual method has questions.
Mm-hmm. Okay, thank you.
But mechanistically, we don't understand it.
Okay.
Okay.
All right. Next question.
Right. Okay. Firstly, I'd like to thank you for your significant contribution to the field. I very much enjoyed your talk, and I also resonate with you. I mean, you're talking about the river flow. You're talking about aging as an attractor, isn't it? Mm-hmm. Aging is an attractor.
Yes.
I mean, we tend to go towards it, but the trajectory that we need to take can differ, right? You say entropy is increasing with aging. Many people talked about it, right? The disorder is increasing as we age. Right? So if you want to extend life long, you can't avoid the attractor, but the trajectory that we take to the attractor can be tweaked so it takes a longer time to reach the attractor. Right?
Well, in some cases, we can avoid the attractor by stopping the aging process. I think it's possible under certain conditions, certain levels of biological organization. Like, for example, Hydra, there are stem cells that can produce any other cells, so cells constantly replaced. But in mammals, it's not possible because we have non-renewable cells and structures. Like we have neurons or cardiomyocytes, we have skeleton, we have eyes. This already means that aging cannot be stopped in mammals.
So my question to you is actually, okay, so we have many drugs already, like you mentioned rapamycin, metformin. The mechanism of how it does it is not very clear, right? And when we talk about entropy, we're not talking about single molecule. We're talking about a network of molecules. So when we want to develop a drug that extends age, we should look at entropy, how it regulates entropy, but should we look more at the molecular level? Like so what are the molecules we need to target to get that? So that's my question.
I really haven't discussed any of that. I try to go back to the very basic, right? But then once you go from damage to biological organization, there are so many different ways. So depends on what you'd like to target with the drug. Are you going to target diseases or like functional decline or mortality or damage accumulation? It depends on that and how organisms is organized. You would have different strategies, right? So it's a really whole separate question, I would say.
I'll talk to you.
Okay,
Thank you.
yeah. All right, let's move quickly to the next one, the next question.
Thank you very much for your work. Concerning the ground zero, once again. So, you say that aging clocks we can use, well, you used a lot of different clocks, but still, how do you think, is there some mechanistic approach that can be used to show some rejuvenation in these states? I mean, the cells are still there, the proteomes are still there. Like, can we do something better than clocks?
We are trying. There are ongoing projects in the lab, but we don't have clear understanding. We don't have it. So I wish I would tell you how this happens. I don't know.
Okay, thank you.
All right, next one.
Thank you for the talk. Very quick question, and philosophical one. Do you actually believe that radical life extension 2 times or more to 200 years, 300 years is possible? Regarding the old discussion we have these 2 days.
In humans.
In humans.
And in humans with the current technologies and methods, I don't think so. So we don't have any, like, all of the tools we currently have, like any lifestyle drugs, simple genetic manipulations.
Not currently, but potentially.
Well, theoretically, of course, it's possible. but in a practical sense, with the current tools, no. There was already discussed at conference, and our data suggests the same. There are maximal lifespan in humans. So it can be discussed where it's like 105 or 140 or so, but in that range. And this kind of encompasses all the simple interventions and manipulations that we know. But in the future, it could be, I don't know, reprogramming or organ replacement or some other technologies. It may be possible, but not now.
Okay, maybe one last question.
Thank you for your talk. I was wondering, in the bit where you talk about rejuvenation during embryogenesis, have you considered that the egg cell and the sperm are also under significant evolutionary pressure to be the best that the human could approach? So is that part of?
They are very well protected. Their biological age consistently is extremely very low.
But also the eggs that are selected are going to be the better of the batch as well. So surely
they are selected. They are selected, and also there is a selection for sperm as well. But it's just unavoidable. They are live cells. They stay there in the host for decades, right? So they accumulate changes which are slightly deleterious, even though they are exceptionally well protected. They're extremely young. It's just they age slightly, so that means they must be rejuvenated. So that's what we think. Yeah, but how exactly this happens, I don't know. And but the germline itself, the way it develops, it's actually another strategy of biological strategy to export entropy, basically. Yeah. It still has this physical essence.
Okay, thank you very much, Professor Gladyshev. Very insightful talk.